Chapter 19 looks at the reproductive system. So much of this semester we've discussed the human body as an individual identity, but in this chapter we'll discuss the final body system, the reproductive system, and its purpose of propagating the human species. So the first section gives you an introduction to the basic components of the human reproductive system, and then we look at the specifics of the male reproductive system, and in the following section the specifics of the female reproductive system. Then we take a look at the details involved in the physiology of sexual intercourse in males and females, and we conclude the lecture with age-related changes that occur in the reproductive system. So the reproductive system produces, stores, nourishes, and transports gametes, the reproductive cells. In males, it's spermatozoa, or sperm for short, that combine with secretions of accessory glands to form semen. Oocytes in females is an immature gamete, and when fertilized by sperm, it becomes an ovum. Gonads are the organs that produce gametes and hormones. In adult males, the testes secrete androgens and 1/2 billion sperm each day. In adult females, the ovaries release 1 oocyte per month. In both males and females, ducts that receive and transport gametes are connected to chambers and passageways that open to the exterior of the body. Accessory glands and organs secrete fluids into the reproductive system ducts, and the external genitalia are the perineal structures. The next two sections are devoted to articulating the differences between the male and female reproductive system, and we start with the male. So proceeding from each testes, the spermatozoa travel within structures composing the male reproductive tract. The epididymis, the ductus or vas deferens, the ejaculatory duct, and the urethra before the sperm leaves the body via the external urethral orifice. Accessory organs include the seminal gland, the prostate, and the bulbo-urethral gland. The external genitalia of the male includes the penis and the scrotum. The scrotum encloses the testes, the primary male sex organ, and they hang outside of the pelvic cavity. The scrotum contains 2 chambers, the scrotal cavities, and those are seen on the next slide. The scrotum contains 2 chambers, the scrotal cavities, one for each testis. The wall of the scrotum has a thin, smooth muscle layer called dartos, which wrinkles the scrotal surface. The cremaster muscle is a skeletal muscle that pulls the testes closer to the body to ensure proper temperature for sperm maturation. The tunica albucina is a dense fibrous capsule that surrounds each testis and extends into the testis, forming partitions or septa that subdivide the testis into roughly 250 lobules. In the lobules, seminiferous tubulars are the site of spermatogenesis, and more on spermatogenesis in a few slides. But sperm then pass through the ret testis and efferent ductules before reaching the epididymis, the beginning of the male reproductive tract. Looking at the microscopic view of the testis, you can see more detail of the seminiferous tubules. Note that the nerve cells that support the formation of the sperm and the spermatids undergoing spermatogenesis. They're becoming more mature as they near the lumen of the seminiferous tubule. Interstitial cells found between tubules produce testosterone. I mentioned spermatogenesis already, and this is a series of cell divisions pictured here in the seminiferous tubules of the testes and include 3 processes. Spermatogenesis begins with the mitotic division of stem cells called spermatogonia, and these are located in the outermost layers of the cells in the seminiferous tubules. This process, this first mitotic division, produces diploid daughter cells. And notice the term diploid in this figure. In humans, that means 23 pairs for a total of 46. So this daughter cell then is going to differentiate into spermatocytes that undergo meiosis. Now, meiosis is a special form of cell division that produces gametes, and these gametes contain half the number of chromosomes found in other body cells. So they are called haploid. And each of these haploid cells are genetically unique too. So the spermatotids formed in meiosis develop into highly specialized spermatozoa during spermiogenesis, and these spermatozoa enter the fluid within the lumen of the seminiferous tubules. Meiosis has a lot of the same mechanisms as mitosis. In fact, they even have stages that are named very similarly. However, synapsis is a special event that only happens in meiosis, not during mitosis. So this is when crossing over occurs between two pairs of chromosomes and genetic information is exchanged between the tetrads. And this is why the cells that come out of meiosis 1 are genetically unique, whereas the cells that come out of mitosis are genetically identical. The diploid daughter cells then undergo reductive division of meiosis 2, meaning the overall number of the chromosomes is cut in half, so that at the end of meiosis 1 and 2, one diploid primary spermatocyte produces 4 haploid, genetically unique spermatids. So the entire process of spermatogenesis, from spermatogonial division to the release of a physically mature spermatozoon, takes approximately 9 weeks. Each spermatid that comes out of meiosis 2 matures into a spermatozoon. The whole process is supported by the actions of nurse cells. The cytoplasms of these nurse cells surround the developing sperm, which controls the chemical environment. So they provide the nutrients that enter by diffusion from the interstitial fluids, and they secrete hormones that promote the production and differentiation of the spermatozoa. There's another type of hormone that they create. It's also called inhibin. So this, and I'll describe the role of inhibin in a couple of slides. But this is a slide that shows the anatomy of a spermatozoon. It has 3 distinct regions. The head contains the nucleus with chromosomes, and it has an achromosome, a tip that has enzymes to penetrate an oocyte. The middle piece has a lot of mitochondria to provide the energy for movement. And the tail, or the flagellum, moves the sperm from one place to another. We have a short neck that attaches the head to the middle piece and contains the centrioles of the spermatozoon. So though the spermatozoa that leave the testes are not yet capable of fertilizing an ovum, they are still incapable of coordinated locomotion. So cilia will generate currents that carry the spermatozoa to the epididymis. The epididymis is a long, about 7 meters, coiled tube, and takes the sperm about 2 weeks to travel the length of the epididymis. The epididymis adjusts the composition of the fluid that's produced by the seminiferous tubules. It recycles damaged spermatozoa. It provides storage for mature spermatozoa, and it prevents capacitation, which is a process that activates the swimming capacities of the sperm. The Ductus Deferens is also called the vas deferens, and it goes into the pelvic cavity through the spermatic cord, which is a sheath of connective tissue and muscles that encloses blood vessels, nerves, and lymphatics that are serving the testes. And this will pass through the inguinal canal of the body wall, lateral to the urinary bladder, and curve down past the ureters towards the prostate gland. So this is another view of the Ductus Deferens, which is pictured in purple. And notice how it curves down past the ureters towards the prostate gland. This is where the seminal gland ducts merge with the vas deferens and becomes the ejaculatory duct. This is a short passageway that goes into the prostate gland and empties into the urethra. So the urethra is a shared tube that's used for both urine and semen, and it runs from the urinary bladder to the tip of the penis. Next, we'll discuss the accessory glands that produce about 95% of the fluid component of semen, starting with the seminal glands, which are also called the seminal vesicles. It's a tubular gland that's coiled and folded just posterior to the urinary bladder. It contributes 60% of the semen fluid, which contains fructose for metabolism by the sperm, prostaglandins, which stimulate smooth muscles in the tract, fibrinogen, which forms semen clots in the vagina, and slightly alkaline fluids to buffer the acid environment of the vagina. It also provides a fluid that can initiate capacitation. So now, at this point, we do want the sperm to have their mobile capacity. So this is for the development of motility of sperm tails. The prostate gland is a small, muscular, round organ that surrounds the urethra as it leaves the urinary bladder. Prostatic fluid is about 20 to 30% of the volume of semen, and it contains semanoplasmin, which is a protein with some antibiotic properties. It appears to help prevent urinary tract infections. The bulbo-urethral glands are also called the Cowper glands. So these are paired small glands that secrete the final amount of fluid in semen, which is a thick alkaline mucus, again to buffer the acidic environment of the vagina and also to lubricate the glands of the penis. Semen is the fluid that contains sperm and the secretions of the accessory glands of the male reproductive tract. A typical ejaculation contains 2 to 5 milliliters and is expelled by the body and contains spermatozoa in about 20 to 100 million spermatozoa per milliliter. Seminal fluid, which contains glandular secretions, ions, and nutrients for the sperm, and also enzymes like proteases to dissolve the mucus in the vagina and enzymes that clot semen. Next, we'll consider the external genitalia. So the penis introduces semen to the vagina during intercourse. It also conducts urine through the urethra. There are 3 regions to the penis. The root of the penis is the fixed portion connected to the body wall. The body or shaft of penis contains erectile tissues, and the glans is the expanded distal end with the external urethral orifice. The prepuce, which is not shown here, is a fold of skin that covers the glans but is often removed. The erectile tissue of the penis is a maze of vascular channels, elastic connective tissue, and smooth muscle. The anterior of the flaccid penis is covered by 2 corpora cavernosa networks of erectile tissue, and surrounding the urethra we have another erectile tissue called the corpus sponginosum. In the resting state, there is very little blood flow to the arterial branches, so these are constricted, but during an erection, parasympathetic neurons release nitric oxide, which is a vasodilator. So smooth muscles in the arterial walls are going to relax, the vessels dilate, and you'll have an increased blood flow to the penis, which will result in an erection. So let's take a look at regulation of male reproduction, which all starts in the hypothalamus, which releases gonadal-releasing hormone that will trigger the release of follicle-stimulating hormone and luteinizing hormone from the anterior pituitary gland. Follicle-stimulating hormone targets nerve cells to trigger spermatogenesis. Luteinizing hormone stimulates differentiation of the male reproductive tract and initiates sexual maturity, development of secondary sex characteristics as well. Testosterone also plays a role in negative feedback to the hypothalamus to tell it to stop producing gonadotropin-releasing hormone. And the production of inhibin by the nerve cells provides the negative feedback that's needed to the anterior lobe of the pituitary gland to stop secreting follicle-stimulating hormone. So that brings us to the first checkpoint of this chapter and the end of this first recording.